This is the genetic engineering lab. You guys are going to need to read the introduction, and you're going to need to read the steps of the lab, even though I'm going to do it for you. I do want to make sure that you have looked at the genetic engineering slides on Google Classroom so that you know exactly what's happening here in this diagram. If you've not already looked at the genetic engineering slides, I want you to actually stop and go do that first before you continue with this video. So as long as you are aware of the background information, then you know that genetic engineering allows us to take a human gene and insert it into a loop of DNA in a bacterial cell in order to get the bacterial cell to make the protein coded for by the human gene. And in this case, we're going to imagine that this is insulin. So I've got a strip of human DNA, and I'm going to try to find the gene for insulin. And according to the lab, this is the sequence for the gene for insulin. So I'm going to take a look at my DNA, and I'm going to color it red. So I'm looking for TGT, usually starts after a sequence of A's and T's. So here we go. Here we've got a TGT, CAC, TGA, CA, CA, CTGC, TA, CTCGA. There we go. So this is the gene for the protein that we are after today. Now you guys already learned how to turn a gene into protein. Today we're just focused on the gene itself as part of the DNA. So I'm also going to extract a loop of DNA called a plasmid from a bacteria cell. And it says I should color it green, but this one's already colored green. I have a loop of DNA that I've pulled from a bacterial cell. So I've got human gene and I've got bacterial DNA. Restriction enzymes are used to cut all types of DNA at specific points in base sequence. These enzymes don't just chop the DNA in half, they cut into one strand, then separate the base pairs between strands. Then they cut through the other strand. This process leaves sticky ends of a single strand of DNA where it has been cut. So here, a sequence has been found and the restriction enzyme cuts into this section, but it does so in a way that leaves exposed bases that could pair up with their matching base pairs. They're called sticky ends. In this case, here we have a T, T, A, A, and on this side we have an A, A, T, T. They used to be connected, now they're separated. Using the restriction enzyme template, determine the location on the plasmid DNA that matches the restriction enzyme. So I am looking for a section that looks like this, AATT on the top side, TTAA on the bottom side. So I'm going to look through, I'm looking for AATT. The restriction enzyme would do this automatically because bases have a sort of a static electrical type attraction. Ah, there it is. So my restriction enzyme is actually going to cut right into this like that. Then I'm going to look for the same sequence on my human DNA, and here is actually already been found one of them. The restriction enzyme is going to cut right along there. And there's got to be another one here somewhere, I'm sure of it. It's only 4 bases. There's a good chance that I'll find it. There we go. So now my restriction enzyme can cut in exactly that sequence to cut at a matching place on the 2 pieces of DNA. So it leaves some sticky ends on one sticky end there. And a sticky end there. Now, these sticky ends may be attracted to each other and they can match up. So there's another enzyme involved called ligase. Guess what ligase does? Ligase is going to help bind these sticky ends together. So I'm going to take some ligase enzyme and I'm going to reattach this DNA like that. That pairs nicely. And then I'm going to reform this plasmid loop and bond the other side as well. And attach it with some ligase. Now I have formed a new loop of DNA, a plasmid, that can go right back into the bacterial cell. This newly recombined piece of DNA is now called recombinant DNA. So this recombinant DNA plasmid can go back inside of a bacterial cell, and that bacterial cell will continue about doing its life functions. It will reproduce, and in doing so, it will copy the section of DNA that includes the human gene. It will transcribe and translate its own proteins. So as it's reading its own genes, it will be making its own proteins, and then as it reads the human gene, it will make the human protein. It'll happen automatically, and it won't know, the bacteria won't know the difference between a bacterial DNA and the human DNA. And then we actually would then cultivate the bacteria in huge vats the same way that we would make beer. We are going to encourage this bacteria to grow and to reproduce and to make its own protein. And then when it has made enough, we will extract the protein that it has made, isolating the human insulin and packaging it up into vials. Now, why do we care about this? Well, I'm going to let my husband explain that.
So I'm betting you're thinking that's pretty weird. That, well, that's my pancreas. Yours is located somewhere behind your stomach. About a 6-inch long organ. Produces insulin for you. I'm a type 1 diabetic and my body doesn't produce insulin. So as this primes, we'll do some introductions. I'm Mr. Gates. I am Mrs. Gates' husband, and I'm type 1 diabetic. On this arm, I've got this cool little thing. It makes me all bionic. It does constant glucose monitoring. It measures the sugar that is in my blood. This little thing right here that I'm setting up right now is an insulin pump. And I'm willing to bet that some of you have friends that have them as well. This is a newer one. Instead of being attached to my stomach, I can put it on my arm. And I'm actually going to put it on my other arm opposite the glucose monitor. So all this fun little process, and I get to do this once every 3 days or so. Fun little thing. It's got this whole sticky area. I'll just pop it on there. It has some cool little glue. This is the coolest hospital glue I've ever seen. This stuff sticks so great. Sticks. I can go swimming with it. Can stand out in the rain, the snow, because it's snowing today in the middle of April. Yay! Bet y'all love that. Okay. So this device here gets everything going, and it works on Bluetooth. So I'm going to get it started, and it's going to insert the needle into my skin. And whoa! Oh no, I'm just joking. I'm kind of used to it. And there it's priming, and everything's in and set to rock. And with monitoring and staying active, eating healthy, I'll be healthy because of this cool little machine. Have a good one.